Hey dreamers, night owls, and curious minds. Welcome back to Hafee Sleep, your cozy corner for fascinating stories that unwind your mind and spark your imagination. I'm Hafee, and tonight's journey is truly special. Imagine holding life's entire instruction manual and learning how scientists discovered a way to edit it. We're diving into one of biology's greatest breakthroughs, born from a microscopic war between bacteria and viruses that changed science forever. But before we begin, if you love stories that blend science, wonder, and a dash of magic, hit subscribe. New videos drop every week to soothe your curiosity. Tap the like button if you're excited to explore how tiny bacteria gave us a tool to rewrite DNA. Tell me in the comments. Where are you watching from right now? Late night in London? Early coffee in Tokyo? Your location plus time makes our community feel wonderfully alive. Truth? Your comments light up my day. Knowing you're out there, wrapped in blankets, sipping tea or stargazing, makes these stories matter. Let's begin. Picture this. You're holding the most intricate, complex, mind-bogglingly detailed instruction manual ever written. It's not for building a wardrobe from a certain Swedish store, nor is it for programming the latest smartphone. No, this manual tells a single cell, the tiniest building block of life, how to build and maintain an entire living thing. A blade of grass, a towering oak, a fluttering butterfly, a curious dolphin, you, me. Every living thing on this planet carries its own unique version of this manual within almost every single cell. This manual is called the genome, and it's written in the astonishingly elegant four-letter code of DNA. Now imagine if we could not just read this manual, but actually edit it, correct a typo here, adjust a confusing instruction there, maybe even add a whole new beneficial paragraph. That, my friends, is the revolutionary, exhilarating, and slightly mind-bending world of genome editing. It sounds like pure science fiction, doesn't it? Like something out of a comic book or a futuristic movie. But hold on to your hats, because this isn't fiction anymore. It's happening right now, in laboratories buzzing with activity all over the globe. Scientists are peering into the very heart of life, wielding molecular tools with incredible precision, learning to rewrite the code that shapes biology itself. It's a power that feels almost godlike, and like any immense power, it comes wrapped in layers of dazzling potential, profound ethical questions, and a fair dose of healthy caution. So, let's pull up a chair, metaphorically speaking, and dive deep, really deep, into the ins and outs of genome editing. We'll explore what it truly is, the brilliant minds who paved the way, the ingenious molecular machines that make it possible, the breathtaking benefits it promises, and the significant concerns it rightly raises. We're going to unpack it all, piece by fascinating piece, in language as clear and friendly as a chat over coffee. Don't worry about the journey being long. Think of it as an epic adventure into the microscopic heart of life itself. Ready? Let's begin at the very beginning, a very good place to start. Before we can talk about editing, we really need to understand what we're editing. So let's talk about the genome. Imagine your body is a phenomenally complex city. Trillions of citizens, your cells, working nonstop. There are power plants, mitochondria, transportation networks, blood vessels, waste disposal systems, liver, kidneys, communication lines, nerves, and construction crews constantly repairing and rebuilding. Now, how does this entire city know how to function? How does a single brick, a cell, know whether it's part of a skyscraper, a bone, or a cozy cottage, a skin cell, or a bustling factory, a liver cell? How does it know when to grow, when to divide, when to produce specific chemicals, when to call for help, when it's time to retire? The answer lies in the central planning office, the master blueprint held securely within the nucleus of almost every single cell. That blueprint is the genome. The genome isn't one giant document. It's more like a vast library. The books in this library are called chromosomes. Humans have 23 pairs of these chromosomal books, neatly stacked in the nucleus. If you were to take all the DNA from one human cell and stretch it out, it would be about 2 meters long. But it's packaged with incredible efficiency, wound around special proteins like thread on a spool, so it fits into a space way, way smaller than the head of a pin. Now, the text within these chromosomal books is written using a remarkably simple alphabet. Just 4 chemical letters. A for adenine, T for thymine, C for cytosine, G for guanine. That's it. The entire instruction manual for building and running a human being. Your eye color, your potential height, your susceptibility to certain diseases, the basic shape of your nose, is written using endless combinations of just these 4 letters. A, T, C, G. Think of them like binary code, zeros and ones for computers, but with 4 options instead of 2, making it vastly more complex and capable. Specific sequences of these letters form meaningful units of instruction called genes. A gene is essentially a recipe, usually for making a specific protein. Proteins are the workhorses of the cell and, by extension, your entire body. They are the bricks and mortar, like collagen in your skin. The machines, like the enzymes that digest your food. The messengers, like hormones. The transporters, like hemoglobin carrying oxygen in your blood. And the defenders, like antibodies. Almost everything your body does, from blinking to thinking, relies on proteins doing their specific jobs. So, a gene holds the code that tells the cell how to assemble a particular protein, amino acid by amino acid. But genes aren't just strung together like beads on a necklace. The genome is far more complex and sophisticated. Only about 1 to 2% of the human genome actually codes for proteins. That's the part with the direct recipes. The rest? Well, for a long time, it was dismissively called junk DNA. We now know that's a huge misnomer. This non-coding DNA plays crucial regulatory roles. Think of it like the control systems, the punctuation, the formatting, and the indexing of the genome library, promoters. These are like "start here" signs, telling the cellular machinery where to begin reading a gene. Enhancers, silencers. These are like volume knobs or on-off switches, located sometimes far away from the gene itself, boosting or reducing how much of a particular protein is made. Introns. These are sections within a gene that get transcribed, copied initially, but are then cut out before the protein is made. They might play roles in regulating gene activity or allowing for different versions of a protein to be made from the same gene. Alternative splicing. Telomeres. These are protective caps on the ends of chromosomes, like the plastic tips on shoelaces, preventing the DNA from fraying and getting damaged each time a cell divides. Centromeres. These are special regions that help chromosomes line up correctly when a cell divides. Regulatory RNAs. Vast stretches code for RNA molecules that don't make proteins but instead help control which genes are turned on or off and when. So, the genome is this dynamic, three-dimensional, exquisitely regulated library of information. It's not static. It's constantly being read, interpreted, and responding to signals from inside and outside the cell. Genes can be turned on or off, a process called gene expression, in complex patterns depending on the cell type, the stage of development, environmental cues, and health status. A skin cell has the same genome as a liver cell, but it reads and uses a completely different set of pages from the instruction manual. That's the magic and complexity of it all. The idea of deliberately changing an organism's genetic makeup, its genotype, to alter its observable characteristics, its phenotype, isn't new. For millennia, humans have been doing a crude form of genetic manipulation called selective breeding. Think of farmers choosing the plumiest plum or the sweetest apple from a tree and planting its seeds, hoping the next generation will be even better. Or think of dog breeders selecting specific traits to create the vast array of breeds we have today, from tiny Chihuahuas to giant Great Danes. This works because you're indirectly selecting for combinations of genes that produce the desired traits. But it's slow, imprecise, and limited to mixing and matching the genes already present within a species or closely related species that can interbreed. The dream of precise targeted editing, going directly into the DNA library, finding a specific sentence on a specific page of a specific book, and changing a single letter, that required a deeper understanding and new tools. So we left off talking about that incredible instruction manual, the genome, tucked away inside almost every single one of your cells. We know it's written in a surprisingly simple alphabet, just A, T, C, G, but arranged in mind-bogglingly complex sequences that tell your body how to be you. But here's a question that's probably gnawed at brilliant minds for decades before the big breakthrough. How? How on earth does this molecule, DNA, actually work? How does it store all that intricate information? How does it copy itself so faithfully when a cell divides, making sure every new cell gets a perfect copy of the manual? And crucially, for our story about editing, how could it possibly be changed? Imagine trying to fix a fantastically complex machine without even knowing what it looks like or how its parts fit together. That's where science was stuck with DNA for a long time. They knew it was important. Incredibly important, the stuff of genes. But its physical structure, its actual shape in three-dimensional space, was a complete mystery. Without knowing that structure, understanding how it functioned, let alone dreaming of editing it, was like trying to read a book in the dark, blindfolded, with gloves on. Frustrating, right? The monumental leap, the absolute game changer that lit the fuse for everything that followed, including our modern genome editing marvels, was the discovery of DNA structure. Not just any structure, mind you, but the now iconic double helix. Picture a twisted ladder, or maybe a spiral staircase, gracefully winding upwards. That's the double helix. And the story of how it was figured out is a tale of brilliant minds, intense competition, overlooked contributions, and one of the most beautiful aha moments in scientific history. It happened in 1953, primarily credited to James Watson and Francis Crick, working at the Cavendish Laboratory in Cambridge, England. But, like any good detective story, the clues came from many sources, and the full picture involves key players whose contributions were initially overshadowed. Think of it this way. DNA was the ultimate safe, holding life's deepest secrets. Scientists knew the safe existed. They knew it contained unimaginable treasures, the genetic code. They even knew some of the components it was made from, the chemicals adenine, thymine, cytosine, guanine, our ATCG. But they had no idea what the lock looked like or how the mechanism inside worked. Watson and Crick weren't necessarily the ones doing the most direct experiments to probe the safe. Instead, They were master puzzle solvers, brilliant model builders. They gathered crucial clues from others and assembled them into the key that finally opened it. One of the most vital clue givers was Rosalind Franklin, an exceptional chemist and X-ray crystallographer working at King's College London. Franklin was a master at a technique called X-ray diffraction. Imagine shining a powerful beam of X-rays through a crystal of a substance. As the X-rays hit the atoms inside the crystal, they get scattered in specific patterns. It's like throwing pebbles into a pond with obstacles. The ripples interfere with each other, creating a complex pattern of waves. By painstakingly analyzing these patterns, essentially the shadow cast by the molecule's atoms, experts like Franklin could deduce the molecule's shape, the angles between its bonds, and how its atoms were arranged in space. It was incredibly difficult work, requiring immense skill, patience, and mathematical insight. Franklin focused her powerful skills on getting the clearest possible X-ray diffraction images of DNA fibers. Her work was meticulous. In May 1952, she captured an image of exceptional clarity, known as Photograph 51. This wasn't just any blurry picture. It was a striking, clear X pattern with distinct dark marks at regular intervals. To the trained eye, this pattern screamed helix. More specifically, it strongly suggested a double helix structure, with the strands running in opposite directions. Franklin, a cautious and rigorous scientist, was methodically analyzing her data and hadn't yet published her definitive conclusions. Meanwhile, over in Cambridge, Watson and Crick were racing to build physical models of DNA. They were trying to piece together the jigsaw puzzle using known chemical facts. The sizes and shapes of the A, T, C, G molecules, called nucleotides, how atoms like to bond with each other, and crucially, information about the ratios of these bases in DNA discovered earlier by Erwin Chargaff. Chargaff had found something fascinating and oddly specific. In DNA, the amount of adenine, A, always equaled the amount of thymine, T, and the amount of guanine, G, always equaled the amount of cytosine, C. So, A, T, and G, C. This Chargaff's rule was a massive clue, hinting at some kind of specific pairing between these bases. Watson and Crick also had access to some of Franklin's data, including crucial measurements derived from her work, and, controversially, shown Photograph 51 by Franklin's colleague Maurice Wilkins, without her explicit knowledge or consent at the time. Wilkins, also working on DNA at King's, shared a more collaborative, if sometimes strained, relationship with the Cambridge duo. Franklin's data provided the critical measurements they needed: the diameter of the helix, the distance between the turns, the fact that there were likely two strands. Armed with Chargaff's ratios, AAT, GC, the dimensions from Franklin's X-rays, and their own model-building genius, Watson and Crick had their eureka moment. They realized the structure had to be a double helix. The sugar and phosphate parts of the nucleotides formed the strong, twisting backbone of the latter, the uprights on the sides. Stuck inward from each sugar, pointing towards the center, were the nitrogenous bases, A, T, C, G. But here's the absolute stroke of brilliance, the key that unlocked the safe. These bases paired up specifically across the center, like the rungs of the ladder. Adenine, A, on one strand always paired with thymine, T, on the other strand. Guanine, G, always paired with cytosine, C. And they paired in a very specific way, forming hydrogen bonds, weak, attractive forces between them. A and T formed 2 hydrogen bonds. G and C formed 3. This explained Chargaff's rule perfectly. If A always pairs with T and G with C, then naturally the amount of A equals T, and G equals C in any DNA molecule. More importantly, it revealed the mechanism for copying. Imagine you have your twisted ladder. Now imagine unzipping it right down the middle, splitting the two strands apart. Each strand now has a series of bases sticking out. Because of the specific pairing rules, A only bonds with T, G only bonds with C, each old strand acts as a perfect template. Floating nucleotides in the cell will latch on. Where there's an A on the old strand, only a T will fit. Where there's a G, only a C will fit, and so on. Enzymes then zip up the new strands. Voila! You have two identical double helices where there was only one before. This is semi-conservative replication. Each new DNA molecule consists of one old strand and one brand new strand. It's how every single cell in your body, except red blood cells and a few others, carries an exact copy of your genome, passed down faithfully when cells divide. Think about how fundamental this is for editing. If the sequence of bases is the information, the words in the instruction manual, and copying relies on this precise base pairing, then changing a base sequence, an edit, would be passed on when that cell divides. If you fix a typo in the DNA of a stem cell, all the cells that come from it could carry that fix. The double helix structure revealed the mechanism for both faithful inheritance and the potential for targeted change. But the double helix revealed so much more than just copying. Information storage. The sequence of bases along one strand is the code. It doesn't matter what order the bases are in along the backbone. It's the sequence itself that holds the meaning, just like the sequence of letters forms words. The double helix showed where this information is physically written. Stability, protection. The twisting double helix structure, with the bases safely tucked inside and the strong sugar phosphate backbone outside, protects the precious genetic code from chemical damage. It's a robust design. Accessibility for reading. While the structure is stable, it also needs to be read. The double helix can be locally unzipped by cellular machinery, so that the code on one strand can be transcribed into RNA, the first step in making proteins, while the other strand acts as a backup. This showed how the instructions could be accessed without destroying the whole molecule. The potential for mutation and editing. The structure also hinted at how mistakes, mutations, could happen. If something damaged a base, say, radiation knocks a piece off it, or if the copying machinery slipped up and inserted the wrong base during replication, an A opposite a C instead of a T, for example, that would change the sequence. That changed sequence could alter a gene's instructions, potentially causing disease. Or crucially, it showed that the sequence could be changed. If nature could make mistakes, or if chemicals or radiation could cause changes, then perhaps scientists could find ways to make specific desired changes. The double helix revealed DNA not as an unchangeable stone tablet, but as a dynamic molecule where the information could, in principle, be rewritten. It planted the seed for genome editing. Understanding the structure was like finally getting the blueprints for the safe. Now scientists knew exactly how the lock worked, base pairing, how the contents were arranged, the sequence along the strand, and how duplicates were made, replication using the template. This fundamental knowledge was absolutely essential for everything that came next in molecular biology, especially the development of tools to manipulate DNA. Before 1953, imagining targeted gene editing was pure science fiction. After 1953, it became a conceivable, albeit distant, engineering challenge. Scientists now had a clear target: Figure out how to locate a specific sequence of A's, T's, C's, and G's within the vast genome, and then figure out how to modify that specific sequence deliberately and accurately. The double helix was the foundational map that made navigation possible. It transformed DNA from a mysterious chemical into a comprehensible molecular machine, opening the floodgates to understanding genes, genetic diseases, and ultimately, the very real possibility of rewriting the code of life itself. Watson, Crick, Franklin, Wilkins, Chargaff, they handed biology the key. The journey to learn how to pick the lock with precision had just begun. All right, so we've got the map, the glorious double helix, unveiled in 1953. We understand how DNA stores information in its sequence of A, T, C, G, and how it faithfully copies itself using those strict base pairing rules, A with T, G with C. Fantastic. But knowing how the safe looks and copies its contents is one thing. Actually opening it up, carefully taking out a specific page, maybe modifying it, or inserting a whole new chapter from a completely different book. That's a whole other level of tinkering. That's where the story zooms forward into the wild and woolly 1970s and 80s. Buckle up, because this is when scientists started getting their hands dirty with the actual text of life, and things got seriously revolutionary, even if it was still a bit, well, blunt. Imagine you're a scientist back then. You know genes are made of DNA. You know specific genes do specific things. You desperately want to study a human gene, say the one for insulin, that crucial hormone that keeps our blood sugar in check. But you can't exactly experiment on human genes easily inside a living person. What if you could take just that gene, isolate it, and put it into something simple and easy to grow like a bacterium? Then you could study it, produce tons of it, maybe even make medicine. But how? How do you cut out one specific gene from the vast, tangled library of the human genome? And how do you paste it into the tiny, circular genome of a bacterium so it actually works? Enter the heroes of this era, recombinant DNA technology, also known as genetic engineering. And its pioneers, folks like Paul Berg, Herbert Boyer, and a Stanley Cohen. Their breakthrough was realizing that nature, in its infinite inventiveness, had already invented the tools they needed. They just had to find them and figure out how to use them. The first magical tool they discovered? Restriction enzymes. Think of these as nature's incredibly precise molecular scissors. Bacteria make these enzymes as part of their own little arms race against viruses, bacteriophages. When a virus injects its DNA into a bacterium, the bacterium fights back by chopping up the invading viral DNA. Restriction enzymes are the molecular ninjas that do the chopping. The brilliant thing? They don't just chop willy-nilly. Each type of restriction enzyme recognizes a very specific short sequence of DNA letters, its own unique cut here signal. For example, one famous enzyme, EcoRI, from E. coli bacteria, recognizes the sequence GAATC. It scans along the DNA double helix, and every single time it sees GAATTC in that exact order, it makes a precise cut between the G and the A on both strands. It's like a pair of scissors programmed to only snip through a ribbon whenever it sees the pattern blue, red, green. Suddenly, scientists had a way to cut DNA predictably. They could take human DNA, chop it up with a restriction enzyme, and get a whole bunch of fragments. One of those fragments would contain the insulin gene. Well, theoretically. Finding the specific fragment with the insulin gene among thousands was still like finding a single specific sentence in a library after randomly tearing all the books into pieces. But it was a start. They could also cut open the small, circular DNA molecules that bacteria carry, called plasmids, using the same restriction enzyme. Because the enzyme cuts the same specific sequence in both the human DNA fragment and the plasmid, it creates matching sticky ends. Imagine cutting a ribbon with zigzag scissors. The cut ends have complementary zigzags that want to fit back together. That's what happens with DNA cut by many restriction enzymes. The ends are uneven, sticky, and have exposed bases that want to pair up. E.g., an ATT end from one fragment wants to bond with a TTAA end from another. But sticky ends alone aren't enough for a permanent fix. You need glue. Enter the second magical tool, DNA ligase. This is nature's molecular glue. Its job in the cell is to mend breaks in the DNA backbone, sealing the sugar phosphate chains. Scientists harness this enzyme to stick together the human DNA fragment with the insulin gene and the cut plasmid. The matching sticky ends help the fragments find each other and align correctly, and then DNA ligase comes along and permanently welds the sugar phosphate backbones together. Voila! You've created a recombinant DNA molecule, a hybrid, a chimera, a bacterial plasmid now carrying a human gene. The next step is like inserting a modified instruction manual into a tiny factory. Scientists take these recombinant plasmids and introduce them back into bacteria. This process is called transformation. Some bacteria will take up the plasmid. As the bacteria grow and divide, they copy their own DNA, including the plasmid. And crucially, they read the genes on the plasmid. If you've designed it right, the bacterial machinery recognizes the human insulin gene's instructions, with a little help from bacterial control signals added by the scientists, and starts churning out human insulin protein. It's like convincing a tiny, single-celled organism to start manufacturing a complex human hormone. Absolutely bonkers when you think about it. And this wasn't just a lab trick. This is how we got human insulin produced by bacteria, replacing the older, less ideal insulin extracted from pigs or cows. It was safer, purer, and more reliable. A life-saving medical revolution born directly from this fundamental genetic engineering technique. Think about that next time you hear about insulin. It's likely made by bacteria carrying a little piece of human DNA magic. This was huge, monumental. It proved we could move genes between wildly different species, from animals to bacteria, later from plants to animals, and so on. It opened the floodgates for producing other vital human proteins in bacteria or yeast, growth hormone, clotting factors for hemophilia, vaccines, and more. It transformed biology and medicine. Scientists could now isolate genes, study them, mutate them in controlled ways, and see what happened. But, and there's always a but, as revolutionary as it was, recombinant DNA technology was still, fundamentally, a bit of a blunt instrument. Remember our library analogy? This was like grabbing an entire chapter, or even just a random paragraph, from one book and randomly inserting it somewhere, anywhere, into a different book in the library. You used molecular scissors, restriction enzymes, that cut at specific sequences. Yes, but those sequences might appear many times in the genome, or worse, not at the exact start and end of the gene you wanted. You might get fragments containing parts of genes, or multiple genes. And when you pasted this fragment into the plasmid, or later, directly into an organism's genome, you had very little control over where it landed. The plasmid might integrate randomly into the host cell's DNA, or the fragment might insert itself randomly during the repair process. This randomness caused problems. Disruption. Imagine randomly inserting a new paragraph into the middle of a crucial chapter in your favorite novel. It would ruin the story. Similarly, inserting a new gene randomly into the host genome could land right in the middle of another essential gene, breaking it and potentially harming the cell or organism. This made it risky for therapies. Inconsistent results. Because the insertion site was random, the new gene might land in a genomic neighborhood where it was buried and ignored, silenced, or it might land somewhere where it was turned on way too strongly or at the wrong time. Getting consistent, predictable expression of the new gene was tricky. One batch of engineered bacteria might produce tons of insulin, another, very little, just because the DNA landed in different spots. Lack of precision for repair. This method was great for adding a whole new gene, but what if you just wanted to fix a tiny typo in an existing gene, like changing a single A to a G in a gene that, when misspelled, causes a terrible disease? Recombinant DNA tech, with its focus on cutting and pasting chunks, was far too crude for that kind of microsurgery. It was like trying to fix a single misspelled word by ripping out the entire page and hoping the new page you paste in fits perfectly and contains only that corrected word. Unlikely. The desire for true precision editing, the ability to go to an exact spot in the genome and make a specific, targeted change, whether fixing a single letter or precisely inserting a gene in a safe location, became the next holy grail. Scientists weren't satisfied with random insertions. They wanted the molecular equivalent of a GPS-guided scalpel, combined with a nanopencil and eraser. Enter the homologous recombination era, hoping the librarian is paying attention. Throughout the 1980s and into the 90s, scientists explored various avenues towards this goal of precision. One strategy that rose to prominence exploited something cells do naturally all the time: DNA repair. Cells are constantly fixing nicks, breaks, and mistakes in their DNA. One particularly sophisticated repair pathway is called homologous recombination, HR. Here's how HR works naturally. Imagine one of the two copies of a chromosome in a cell gets badly damaged. Say, a big chunk is broken off. The cell doesn't panic. Well, it does internally, but it has a plan. It uses the other undamaged copy of the same chromosome as a perfect template. Special repair machinery lines up the damaged region with the matching healthy region on the good chromosome. It then meticulously copies the correct sequence from the healthy template over to the damaged spot, effectively photocopying the missing instructions back into place. It's an incredibly precise repair mechanism using the inherent similarity, homology, between the two chromosome copies. Scientists had a brilliant, if somewhat optimistic, idea. Could we trick the cell into using a piece of DNA we provide as the repair template? Instead of relying on the other chromosome, we introduce a custom-made DNA fragment into the cell. This fragment would have two key features: homologous arms, long stretches of DNA sequence on either end that are identical to the sequences flanking the exact spot in the genome we want to edit. Think of it as having the correct page numbers and the first and last sentences of the page we want to replace. The desired change. Sandwiched between these homologous arms is the DNA sequence we actually want to put into the genome. This could be a small correction, fixing a typo, a larger insertion, adding a new gene, or even a deletion, removing a problematic section. The theory was elegant. the cell's HR machinery would recognize the homologous arms on our introduced fragment, match them up with the identical sequences in the genome, and use our fragment as the template for repair. In the process, it would copy our desired change right into the genome at the precise target location. Like leaving a corrected page lying open on the librarian's desk, hoping she notices it matches the damaged page in Book 17, Chapter 3, and decides to use your corrected version when she repairs the book. Possible? Yes. Reliable? Efficient? Not so much, especially in the complex genomes of plants and animals. This technique, gene targeting via homologous recombination, became a powerhouse tool in one particular area: creating knockout mice. Why mice? Because scientists had developed incredibly sophisticated ways to manipulate mouse embryonic stem cells, ES cells. These are special cells from early mouse embryos that can develop into any cell type in the body. Here's the basic knockout mouse recipe. Design the construct. Scientists design the DNA fragment with long homologous arms, matching the mouse gene they want to disrupt, knock out. And in the middle, they insert a cassette that breaks the gene, often a gene for antibiotic resistance, which also serves as a selection marker, into the stem cells. They introduce this fragment into millions of mouse embryonic stem cells growing in a dish. They use methods like electroporation, zapping the cells with electricity to make their membranes temporarily leaky, to get the DNA inside. The waiting game and selection. In a tiny, tiny fraction of these cells, we're talking maybe 1 in a million, the HR machinery actually uses the introduced fragment correctly. It replaces the normal copy of the target gene in the cell's DNA with the modified version containing the disruptive cassette. Scientists then use antibiotics to kill all the cells, except those that took up the construct, which are now antibiotic resistant. Finding the rare cells where HR actually happened correctly at the right gene among thousands of genes is like finding a specific needle in a stack of needles. Making the mouse. Once they identify these rare, correctly edited stem cells, they inject them into a very early mouse embryo, a blastocyst. This embryo is then implanted into a surrogate mother mouse. The resulting pup will be a chimera, a mix of cells from the original embryo and the edited stem cells. If the edited stem cells contribute to the sperm or egg cells of this chimera, then when it's bred, some of its offspring will inherit the edited gene in all their cells. A true knockout mouse, lacking the function of that specific gene. Studying the knockout. Scientists then study these mice intensively. What goes wrong when this gene is missing? Does it affect development, behavior, susceptibility to disease? This tells them what the gene normally does. Knockout mice revolutionized biology. They provided unparalleled insights into the function of thousands of mammalian genes, modeling human diseases and testing potential treatments. It was a triumph of persistence and ingenuity. The catch: why wasn't this the genome editing revolution? As powerful as homologous recombination was for research, especially in mice, it had major limitations that prevented it from becoming the universal genome editing tool everyone dreamed of. Brutally inefficient. That 1 in a million success rate wasn't an exaggeration for many cell types, especially human cells or plant cells. Getting the HR machinery to use your supplied template instead of the cell's own chromosome copy was incredibly rare. It required selecting through millions of cells to find the few correctly edited ones. This made it slow, laborious, and expensive. Forget about editing inside a living human body. You had to work with cells in a dish, painstakingly find the edited ones, and then try to get them back into the body. Only works well in specific cells. It relied heavily on using special cells like mouse embryonic stem cells, which are immortal in culture and easy to grow and manipulate. Most human cells, especially the mature cells you'd want to fix in a patient, like neurons, muscle, or liver cells, are terrible at HR. They either don't do it much, or they favor the quick and dirty repair method, which we'll get to later. Plants were also very challenging. Requires long, homologous arms. To get even that tiny bit of efficiency, you needed very long stretches of matching DNA sequence on either side of your edit, hundreds or even thousands of base pairs. Designing and making these long, perfect homologous fragments was technically demanding and added to the complexity and cost. Not great for simple fixes. While you could design a template to fix a single letter typo, the inefficiency made it utterly impractical for therapies. The effort required to fix one tiny error was colossal compared to the blunt force of adding a whole gene randomly via older methods. So homologous recombination gave us knockout mice and proved targeted editing was possible, in principle, even in complex mammals. It was a vital stepping stone, a proof of concept. But it was like trying to perform delicate watchmaking with oven mitts on. It worked for specialized tasks in ideal conditions, but it was far too clunky, slow, and inefficient for widespread use, especially for the dream of precise human gene therapy or rapid crop improvement. The frustration was palpable. The stage was set. The scientific world was primed, desperately needing a tool that was precise like homologous recombination, but as easy to use and versatile as the molecular scissors of recombinant DNA. They needed something programmable, efficient, and adaptable. The pressure was building. The revolution wasn't just coming. It was brewing in the most unexpected of places, in the ancient immune systems of humble bacteria. But that, my friends, is the story of CRISPR, and we'll dive into that microbial magician in a while. For now, let's just appreciate the ingenious, if sometimes frustrating, tinkering that laid the groundwork, the molecular scissors and glue that started it all, and the hopeful, patient librarians trying to fix one page at a time. Well, buckle up, because the revolution came. And it came from the most unexpected place imaginable, the ancient, ongoing microscopic war between bacteria and viruses. Forget high-tech labs for a moment. Picture instead a bustling, chaotic, trillion times magnified scene in a drop of pond water, or maybe even in your own yogurt. This is where our heroes, and the ultimate tool, were forged. Meet the bacteria. Simple, single-celled workhorses of life. They're everywhere, crucial for everything from digestion to decomposition. But they have enemies, ruthless, efficient, submicroscopic predators called bacteriophages, or just phages for short. Think of phages as nature's ultimate nanodrones. They look like creepy lunar landers under the electron microscope. Their mission? Find a bacterium, latch on, inject their genetic material, DNA or RNA, straight into the bacterial cell, and then hijack the bacterium's own machinery. The hijacked bacterium becomes a helpless factory, churning out hundreds of new phages until, pop, the cell bursts open like an overfilled water balloon, releasing the new phage army to go hunt more bacteria. It's a brutal, efficient cycle. For a bacterium, a phage attack is an existential threat. Now imagine you're a bacterium. These phage attacks are constant. How do you survive? Over millions of years, some bacteria evolved a truly ingenious defense system. It's adaptive, meaning it learns from past attacks. It remembers specific enemies, and it's incredibly precise. This system is called CRISPR. C-R-I-S-P-R. Yes, it's an acronym, a bit of a mouthful. Clustered regularly interspaced short palindromic repeats. Don't let the jargon scare you. We'll crack this code just like scientists crack the system itself. Let's break it down step by step, just like the bacteria do when fighting off an invasion. Step 1. Surviving the attack. Taking a souvenir. Imagine a brave bacterium survives a phage attack. Maybe its defenses were strong, or maybe it just got lucky. Instead of just breathing a sigh of relief and moving on, this clever bacterium does something remarkable. It captures a little piece of the invading phage's genetic material, a tiny snippet of the enemy's DNA code. Think of this like a soldier grabbing a piece of the enemy's uniform or a shred of their battle plans after surviving an ambush. It's a physical piece of evidence from the attacker. Step 2. Filing the mugshot, building the CRISPR library. Now, the bacterium takes this captured snippet of viral DNA and files it away for future reference. But where? It inserts this snippet into a very special section of its own genome. This section is the CRISPR array. Picture this array as a dedicated scrapbook or a most wanted wall inside the bacterium's central command. What makes this scrapbook special is its structure. Repeats. These are short, identical chunks of DNA sequence that appear over and over again, like the same decorative border on every page of the scrapbook. They're often palindromic in a DNA sense, meaning the sequence reads the same forwards and backwards on the complementary strands, like race car, but made of A, T, C, G. This palindromic nature is key for how the system works later. Spacers. Sandwiched between. These repeating borders are the unique snippets, the captured pieces of viral DNA. Each snippet is a spacer. This is the regularly interspaced part. One repeat, then a spacer, viral snippet, then another repeat, then another spacer from a different virus, and so on. Over time, as the bacterium survives more attacks from different phages, its CRISPR array grows, filled with these unique spacers. It's a chronological library of past invaders, a molecular rogues gallery. Each spacer is a unique mugshot of a specific virus the bacterium or its ancestors have encountered. Step 3. Arming the guards. Making the wanted posters. Okay, so the bacterium has this scrapbook of viral mugshots, spacers, neatly filed between its repeat borders. How does it use this to fight future attacks? It needs to turn this static library into an active defense force. Here's how. When the bacterium senses danger, chemical signals hinting at another phage invasion, it flips open its CRISPR scrapbook and makes a working copy. It transcribes the entire CRISPR array into a long strand of RNA. RNA is DNA's close chemical cousin, often acting as a messenger or a working copy of genetic instructions. This long RNA strand contains all the repeats and all the spacers in order. But a long strand isn't very useful for precise targeting. So, the bacterium chops up this long RNA into smaller pieces with the help of special enzymes, often Cas proteins themselves or dedicated helpers. Each small piece consists of one spacer sequence, the viral mugshot, flanked by bits of the repeat sequence on either end. These small processed RNA pieces are called CRISPR RNA, CRRNA, or more commonly for our editing tool, guide RNA, gRNA. Each gRNA is essentially a single wanted poster featuring one specific viral mugshot, the spacer, and the recognizable border, parts of the repeat. Step 4. Seek and destroy, the guided missile launch. Now comes the truly elegant part. each gRNA doesn't float around alone. It teams up with a powerful partner, a CRISPR-associated Cas protein. There are many types of Cas proteins, but the rock star, the one that changed everything for genome editing, is Cas9. Think of Cas9 as a highly trained, incredibly precise molecular bodyguard or assassin. Cas9 has one key talent. It can cut DNA. Specifically, it can slice through both strands of the DNA double helix, a clean break called a double-strand break. But Cas9 has a problem. It's blind. It doesn't know which DNA to cut. It needs a guide. That's where the gRNA comes in. The gRNA binds tightly to Cas9, the part of the gRNA that contains the unique viral spacer sequence, the mugshot, sticks out, exposed. Cas9, now coupled with its gRNA guide, patrols the bacterial cell. The gRNA acts like a super smart homing device combined with a facial recognition scanner. It scans any free-floating DNA it encounters. It's looking for a sequence that perfectly matches its spacer mugshot. Not kind of similar, not almost the same. Perfectly complementary. Base pair for base pair. A to T, C to G. When it finds a stretch of DNA that is the exact mirror image of its spacer sequence, it latches on tightly. This perfect match signals Cas9. Target acquired. This is the enemy. Cas9 then swings into action. It unleashes its molecular scissors, making that precise double-stranded cut right at the spot, specified by the gRNA. It slices the invading phage DNA into pieces, chopping up the enemy's battle plans before they can be used. The phage invasion is neutralized. The bacterium lives to fight another day. It's an adaptive, programmable immune system at the molecular level. From bacterial defense to global revolution, the aha moment. For microbiologists studying this in the late 1990s and 2000s, like the persistent Francisco Mojica in Spain, who first noticed and named these strange repeating patterns in bacterial genomes, this was fascinating biology. Understanding how bacteria fight viruses is cool. Then teams like the one led by Woma Felipe Horvath at Danisco, a company deeply invested in bacterial cultures for yogurt and cheese making, cultures constantly threatened by phage infections, demonstrated experimentally around 2007 that CRISPR was indeed this adaptive immune system. Protecting their bacterial factories from viral sabotage was a huge industrial incentive to figure this out. But the world-changing leap came when a few brilliant minds looked at this bacterial security system and saw something else, the ultimate programmable molecular tool. Jennifer Doudna, a biochemist at UC Berkeley, and Emmanuel Charpentier, a microbiologist then at Umeå University in Sweden, were studying the CRISPR system in the bacterium, Streptococcus pyogenes, the one that causes strep throat. In a flash of insight, they realized the sheer revolutionary potential. They thought, what if the mugshots, spacers, aren't just records of past viral attacks? What if we could design our own mugshots? The bacterial system uses spacers derived from viral DNA. But DNA is DNA. The sequence is what matters. Doudna, Charpentier, and their teams realized that if they could synthesize an artificial piece of RNA that mimicked the gRNA structure, essentially an artificial wanted poster, but instead of a viral sequence, they put in any sequence they wanted. Then Cas9, guided by that artificial gRNA, would hunt down and cut any matching DNA sequence they programmed it to find. Not just viral DNA, any DNA, human DNA, mouse DNA, rice DNA, anywhere. In 2012, they proved it in a test tube. They took the Cas9 protein from strep bacteria, chemically synthesized a custom gRNA designed to match a specific spot on a piece of harmless plasmid DNA, mixed them together, and Bam! Cas9, obediently following its artificial guide, found the exact spot and cut the DNA precisely where predicted. It was a breathtakingly simple concept, executed flawlessly. The implications were staggering. They had created a programmable DNA cutting machine. You change the 20-letter address in the gRNA, and Cas9 goes to a completely new location in the genome. It was like finding a universal lockpick that could be instantly reprogrammed for any lock just by telling it a new key code. This was the eureka moment that ignited the revolution. Doudna and Charpentier rightly won the Nobel Prize in Chemistry in 2020 for this transformative discovery. Meanwhile, almost simultaneously, Feng Zheng and his team at the Broad Institute were racing down a parallel track. They had the same brilliant insight: repurpose CRISPR-Cas9 for editing. Crucially, Zhang focused on the next vital step. Does this work inside living, complex cells? He took the engineered CRISPR-Cas9 system, Cas9 protein plus synthetic gRNA, packaged it up, and delivered it into living mammalian cells, including human cells growing in a dish. And it worked. Not only did Cas9 cut the target DNA inside these complex cells, but the cell's own repair machinery kicked in, allowing Zhang to demonstrate actual gene editing, disrupting genes in a targeted way. This was the crucial proof that CRISPR wasn't just a test tube trick. It could function in the messy, complex environment of cells relevant to human medicine and biology. Zhang's rapid publication cemented CRISPR's potential as a practical editing tool. The impact was immediate and explosive. Suddenly, everything changed. Easy. Designing a new gRNA is incredibly simple. You just need to know the 20 or so letters of DNA sequence flanking the spot you want to cut. Sophisticated software does the rest, ensuring it's unique. You can literally type the sequence into a website, and a company will synthesize and ship you the gRNA within days. Fast. Experiments that took years with homologous recombination, like making a specific mutation in a cell line to study a disease, could now be done in weeks, sometimes days. Creating a knockout mouse while still involving stem cells became significantly faster and more efficient. Cheap. Compared to the laborious, expensive process of making custom proteins for older editing tools, which we'll touch on in a moment, synthesizing a short gRNA is dirt cheap. A simple CRISPR experiment can cost just hundreds of dollars in reagents, putting it within reach of almost any molecular biology lab, even small university labs or labs in developing countries. Versatile. It works in virtually any organism: bacteria, yeast, plants, wheat, rice, tomatoes, insects, fruit flies, mosquitoes, worms, fish, zebrafish, frogs, mice, rats, primates, and of course, human cells. The same basic principle applies everywhere. Multiplexable. You can target multiple genes at once. Just introduce several different gRNAs along with Cas9. Want to knock out 3 genes simultaneously to see how they interact? No problem. This was incredibly difficult or impossible with older methods. Labs around the world exploded with activity. Projects that were pipe dreams became feasible. Want to understand the function of thousands of genes? Do a genome-wide CRISPR screen using a library of gRNAs targeting every single gene and see which knockouts affect cell growth, drug resistance, or disease processes. Want to correct the mutation causing sickle cell disease in patient-derived stem cells? Design a gRNA to target the exact mutation, provide a corrective DNA template, and hope the cell uses it to fix the error. The possibilities seemed endless. CRISPR wasn't just a tool, It was a tidal wave transforming biology, medicine, and agriculture overnight. Before we get completely swept away by CRISPR mania, it's important to tip our hats to the technologies that paved the way and are still valuable players. TALENs and ZFNs. These were the sophisticated, custom-made lockpicks used before CRISPR became the reprogrammable universal key. Remember the desire for precision before CRISPR? Scientists were making progress, but it was hard, expensive engineering. Both ZFNs, zinc finger nucleases, and TALENs, transcription activator-like effector nucleases, work on a similar core principle. Fuse a DNA binding domain, the part that finds the specific address, to a DNA cutting domain, usually the same Foci enzyme used in some restriction enzymes. ZFNs, zinc finger nucleases. Imagine a protein module shaped like a finger. Each zinc finger module recognizes and binds to a specific three-letter DNA sequence, e.g., GCA or TGG. By stringing several different zinc finger modules together like beads, you could theoretically create a custom protein that binds a longer unique DNA sequence, e.g., 4 fingers, 12 letters. Attach a DNA cutting domain to this custom string of fingers, and you have a ZFN that should find and cut that specific 12-letter address. It was a brilliant concept and worked, but it was tough. The catch: designing fingers that bind only their intended triplet sequence reliably and making sure the string of fingers works together without interfering was a complex protein engineering nightmare. It was slow, incredibly expensive, often costing tens of thousands of dollars per custom ZFN pair, and not always reliable. Getting it right felt like crafting a bespoke, hand-filed key for a single specific lock. Powerful when it worked, but not scalable or easy. TALENs, transcription activator-like effector nucleases. A few years later, scientists discovered a better address recognition system in bacteria that infect plants. These bacteria inject proteins called TALEs, transcription activator-like effectors, into plant cells. Each TALE has a central repeat domain where each individual repeat recognizes just one specific DNA letter, A, T, C, or G. The code linking a specific repeat to a specific DNA base was relatively simple and predictable. This was a huge improvement. How it worked. Scientists could now design a TALE protein by stringing together repeats in a specific order dictated by the DNA sequence they wanted to target. Each repeat in the chain corresponded to one base in the target DNA. Attach the DNA cutting domain and you have a TALEN. It was like building an address finder with modular Lego blocks, each block designed to snap onto a specific letter, A block, T block, etc. Assemble the blocks in the order of your target sequence, add the cutter, and off you go. Better, but still. TALENs were significantly easier and cheaper to engineer than ZFNs. The modular code made design more straightforward. They became popular for specialized applications, especially in model organisms and some cell types. But building the TALE protein chain was still a laborious molecular biology process. Making a new TALEN for a new target sequence took weeks and significant lab work. It was like crafting a sophisticated multi-tool lockpick for a specific lock type. Better than ZFNs, but still required skilled craftsmanship for each new job. And like ZFNs, they often needed to be used in pairs for efficient cutting, adding complexity. This is why CRISPR-Cas9 felt like a lightning bolt. Instead of the agonizing protein engineering required for ZFNs and TALENs, crafting a whole new complex molecular machine for each new DNA target, CRISPR simply required synthesizing a short, cheap piece of RNA, the gRNA. change the 20 base sequence in the gRNA, you've instantly reprogrammed the same Cas9 protein to cut a completely new DNA target. It was like swapping the GPS coordinates on your drone instead of building a whole new drone for every new destination. The simplicity, speed, and cost reduction were revolutionary. CRISPR democratized precision genome editing, putting it into the hands of thousands of labs almost overnight. While TALENs and ZFNs are still valuable tools for specific niches, like edits requiring very long target sites or where minimizing off-target effects is paramount and specific TALEN designs excel, CRISPR-Cas9 became the undisputed workhorse, the versatile tool that changed the rules of the game. The bacterial bodyguards had handed humanity the most powerful molecular scalpel ever imagined. The era of precise genome editing had truly, explosively begun. But as we'll see, wielding this scalpel with precision and responsibility brings its own set of challenges and profound questions. Okay, we've got our programmable molecular scalpel, CRISPR-Cas9 or similar systems. We've designed our custom guide RNA to lead it to the exact spot in the genome we want to change. Now what? How does making a cut actually lead to an edit? The magic lies in harnessing the cell's own emergency repair crews. When Cas9 slices through both strands of the DNA double helix, it creates a double-strand break, DSB. This is a serious emergency for the cell. DNA damage happens all the time, but a clean break across both strands is particularly dangerous. If left unrepaired, it can kill the cell or lead to cancer. So, the cell has evolved two main emergency repair pathways to fix double-strand breaks. Genome editing cleverly hijacks these pathways to introduce the changes we want. Pathway 1. Non-homologous end joining, NHEJ. Imagine the DNA strands are like the two rails of a train track. Cas9 comes along and slices both rails clean through. The cell's panic response is, quick, weld the broken ends back together before the whole train derails. That's essentially what NHEJ does. It grabs the broken DNA ends and fuses them back together. But here's the catch. NHEJ is fast, but incredibly sloppy. It doesn't care about perfectly matching the original sequence, it just wants to stick the ends back together. In the process, it often adds or deletes a few DNA letters, nucleotides, at the cut site. It might insert an extra A or T or chop off a C and a G. These small insertions or deletions are called indels. How is this useful for editing? While indels sound messy, they can be devastating for a gene's function. Why? Remember, genes are recipes written in a specific sequence of A, T, C, G. Changing even one letter can alter the meaning. Adding or deleting letters causes a frame shift mutation. Imagine the gene recipe is a sentence. The cat ate the rat. If you delete the first E, an indel, it becomes THC-ATA TETHR-AT. It's nonsense. The protein made from this garbled recipe will likely be completely non-functional. So, by using CRISPR-Cas9 to make a cut within a specific gene and letting the cell repair it via sloppy NHEJ, we can effectively knock out that gene. We break the recipe on purpose. This is incredibly useful for research, studying what happens when a gene is missing, and potentially for therapy, turning off a harmful gene like one causing a disease. Pathway 2, Homology-Directed Repair, HDR. Now, back to our broken train track. NHEJ is the frantic welder. HDR is the meticulous engineer with the original blueprints. HDR is a much more precise repair mechanism, but it's slower and only active when the cell is about to divide or under specific conditions. Crucially, HDR requires a template. This template is usually the matching chromosome, since most cells have 2 copies of each chromosome, one from mom and one from dad. If one copy is broken, the cell can use the intact copy on the other chromosome as a template to perfectly repair the break. How is this useful for editing? Here's the genius part of genome editing. Scientists can provide an artificial repair template. We design a piece of DNA in the lab that has 2 key features: homologous arms, sequences on either end that exactly match the DNA sequences flanking the cut site. This is like having the correct chapter headings before and after the break. The desired edit. Sandwiched between these homologous arms is the new DNA sequence we want to insert. This could be a correction for a disease-causing mutation, changing a G back to an A, a whole new functional gene, or even just a few letters to subtly alter a protein's function. When we make a CRISPR-Cas9 cut and simultaneously introduce this artificial DNA template into the cell, the HDR machinery can, though not always, grab this template. It uses the homologous arms to find the exact location and then copies the sequence in between, including our desired edit, over into the broken spot. Instead of just patching the break, we've tricked the cell into seamlessly incorporating a specific change we designed. This allows for true gene correction or gene insertion. The critical nuances: it's not magic, yet. This all sounds incredibly precise, and it is molecularly. But there are crucial layers of complexity and challenge. Delivery, getting the editing machinery, Cas9 protein plus guide RNA, and if using HDR, the repair template, into the specific cells we want to edit inside a complex organism like a human, is a massive hurdle. We can't just inject it into the bloodstream and hope it finds the right spot. Techniques include viral vectors, using modified harmless viruses like adeno-associated virus, AAV, that naturally infect cells to carry the editing instructions inside. It's efficient but carries risks, like immune reactions or the virus inserting its cargo in the wrong place. Lipid nanoparticles, LNPs, tiny fatty bubbles that encapsulate the editing components and fuse with cell membranes to deliver them inside. This was famously used in some COVID-19 mRNA vaccines and is being adapted for genome editing therapies. Electroporation, zapping cells with electricity to temporarily make their membranes leaky so the editing components can sneak in. Often used for cells outside the body, ex vivo. Microinjection, painstakingly injecting the components directly into a single cell, like a fertilized egg, using a superfine needle. Off-target effects. This is a major concern. While CRISPR is highly specific, it's not perfect. The guide RNA might accidentally latch onto and cut DNA sequences that are very similar to the intended target, but not identical. This could happen elsewhere in the genome, potentially disrupting a healthy gene and causing unintended consequences, like cancer. Scientists are constantly engineering more precise versions of Cas9 and designing smarter guide RNAs to minimize this risk. On-target, unwanted effects. Even at the correct target site, the editing process itself isn't flawless. When using HDR, the cell might choose the quick and dirty NHEJ path instead, leading to indels at the target site rather than the clean edit we wanted. Or, the HDR process might not incorporate the edit perfectly. Efficiency varies greatly depending on the cell type and the specific target. Mosaicism. If editing is done on a multicellular organism, like an embryo or an adult, it might only work in some cells, not all. The result is an organism with a patchwork of edited and unedited cells, a mosaic. This is undesirable for therapies aiming for a uniform correction. The HDR hurdle. HDR is inherently less efficient than NHEJ and works best in actively dividing cells. Many important cell types for therapy, like neurons or muscle cells, don't divide often, making HDR-based correction very challenging. Developing ways to boost HDR efficiency or finding alternatives is a major research focus. Immune response. Introducing bacterial proteins like Cas9 into human cells can sometimes trigger an immune reaction, potentially attacking the edited cells or causing inflammation. Long-term effects. Safety. What are the consequences of an edit, even a perfectly targeted one, over the entire lifespan of an organism or its descendants? We need long-term studies to ensure safety. So, while the core mechanism of find, cut, and repair is elegantly simple in concept, executing it safely, efficiently, and precisely inside a living, breathing organism is an immensely complex engineering and biological challenge. It's like performing microsurgery with molecular tools inside a constantly moving, self-repairing city. Despite the challenges, the potential benefits of genome editing are so profound, so transformative, that they drive the field forward at an astonishing pace. Let's explore the blossoming fields where this technology is already making waves or holds immense promise, revolutionizing medicine, curing the incurable. Genetic disease correction. This is the holy grail. Imagine correcting the single misspelled letter causing sickle cell disease, cystic fibrosis, Huntington's disease, or muscular dystrophy right at the source, in the patient's own cells. Early clinical trials are showing remarkable promise, particularly for blood disorders like sickle cell and beta thalassemia. Patients have essentially been cured after having their blood stem cells edited outside their body, ex vivo, and then transplanted back. This approach avoids some delivery hurdles and mosaicism concerns. Correcting genes in specific organs, like the liver or lungs inside the body, in vivo, is the next frontier, with trials underway. Cancer immunotherapy. Supercharging. Our immune system has natural cancer fighters called T cells, but cancer is sneaky and can hide from them. Genome editing allows us to supercharge a patient's own T cells. We can edit them to better recognize cancer cells, adding receptors targeting cancer-specific markers. Remove brakes, like the PD-1 receptor that cancer uses to suppress the immune response. Make them more persistent and potent killers. These engineered cells, called CAR T cells, chimeric antigen receptor T cells, have achieved stunning remissions in certain blood cancers, leukemias, lymphomas, where other treatments failed. Genome editing is making CAR T therapy more effective, safer, and potentially applicable to solid tumors. Fighting infectious diseases. Can we edit our own cells to be resistant to viruses like HIV? Or edit immune cells to be better at hunting down infected cells? What about editing the viruses themselves? While complex research is exploring these avenues, more immediately, CRISPR-based diagnostics, like Sherlock and Detector, use the system's targeting ability to detect specific pathogen DNA, RNA incredibly sensitively, promising rapid, cheap tests for diseases. Organ transplantation. The severe shortage of donor organs is a global crisis. Could we genetically edit pigs to make their organs compatible for transplantation into humans? Xenotransplantation. Scientists are using CRISPR to remove pig genes that trigger hyperacute rejection in humans and add human genes that help regulate compatibility. While still experimental and facing immune hurdles beyond just genes, the first pig-to-human heart transplants using edited organs have taken place, marking a historic step. Understanding disease. Beyond direct therapy, genome editing is an unparalleled research tool. Scientists can rapidly create cell or animal models with specific human disease mutations to study how the disease develops and test potential drugs, accelerating discovery, transforming agriculture, feeding the future sustainably. Disease pest resistance, editing crops to be naturally resistant to devastating viruses, fungi, bacteria, or insect pests could dramatically reduce reliance on chemical pesticides. Imagine bananas resistant to the fungus threatening global supplies, or cassava resistant to the virus that causes brown streak disease in Africa. Drought climate resilience. As climate change intensifies, developing crops that thrive with less water or in hotter, saltier soils is crucial. Editing genes involved in water use efficiency, heat shock response, or salt tolerance could create hardier varieties. Improved nutrition, biofortification, enhancing the nutritional value of staple crops could combat malnutrition. Examples include rice with higher vitamin A, golden rice 2.0, though traditionally bred, gene editing could refine it. Wheat with higher iron and zinc bioavailability, or soybeans with healthier fatty acid profiles. Reduced food waste. Editing fruits and vegetables to delay ripening or spoilage, e.g., non-browning mushrooms or potatoes, could significantly reduce the massive amount of food lost after harvest. Animal welfare. Health. Editing livestock for disease resistance, e.g., pigs resistant to PRRS virus, cattle resistant to tuberculosis. Resilience to heat stress or hornlessness, avoiding painful dehorning, could improve animal well-being and farm efficiency. Sustainable production. Editing crops for more efficient nitrogen use could reduce the need for polluting fertilizers. Editing microbes used in fermentation could make food production processes more efficient or create new plant-based proteins. Microbial factories. Editing yeast, bacteria, or algae to become super efficient producers of valuable chemicals, pharmaceuticals, enzymes, or biofuels. This could lead to more sustainable manufacturing processes, reducing reliance on fossil fuels and harsh chemicals. Imagine algae edited to churn out biodiesel, or bacteria producing plastic alternatives that biodegrade easily. Functional genomics. Genome editing allows scientists to systematically turn genes on, off, or introduce mutations in cells or model organisms like mice, zebrafish, fruit flies, to figure out what each gene actually does. This is fundamental to understanding biology, development, and disease. Epigenome. Editing. Beyond the DNA sequence itself, chemical tags attached to DNA, methylation, or the proteins it wraps around, histone modifications, control gene activity. This is the epigenome. New tools derived from CRISPR allow scientists to edit these tags, turning genes on or off without changing the underlying DNA sequence, opening new avenues for research and potential therapy. The shadow side. Navigating the ethical minefield. The power to rewrite the code of life is not without profound risks and ethical dilemmas. As we marvel at the possibilities, we must also confront the potential pitfalls with clear eyes and careful consideration. Unintended consequences. The law of unforeseen ripples. Off-target effects. Revisited. As mentioned earlier, the risk of CRISPR cutting DNA at the wrong place is perhaps the most immediate technical safety concern. An off-target cut in a crucial gene could disrupt its function, potentially leading to cancer or other diseases. While detection methods and improved editors reduce this risk, it can never be entirely eliminated, demanding rigorous testing and long-term monitoring in therapies. On-target complexity. Even a perfectly targeted edit might have unforeseen consequences. Genes often have multiple functions or interact in complex networks we don't fully understand. Pleiotropy. Fixing one problem might inadvertently cause another. For example, the mutation causing sickle cell disease also confers resistance to malaria. Correcting the sickle cell mutation might make an individual more susceptible to malaria in endemic regions. We need deep biological understanding before editing. Mosaicism. Risks. If not all cells are edited uniformly, mosaicism can lead to unpredictable outcomes. In a therapy context, unedited cells might still cause disease, or edited cells might behave abnormally. Ethical quagmires. Playing with the blueprint of humanity. Germline editing. Altering future generations. This is arguably the most controversial aspect. Germline editing means making changes to sperm, eggs, or early embryos. These changes would be incorporated into every cell of the resulting individual, and crucially, would be passed on to all their descendants. While potentially preventing devastating inherited diseases from being passed down forever, the risks are immense. Irreversibility. Once released into the human gene pool, the change is permanent for that lineage. Mistakes or unforeseen consequences cannot be recalled. Consent. Future generations who inherit the edit cannot consent to it. Safety. The risks of off-target effects and mosaicism are even harder to assess in embryos and carry implications for an entire life and beyond. The slippery slope. If we allow editing to prevent severe disease, what about editing for enhancement? Taller stature, higher intelligence, resistance to common illnesses, athletic ability, eye color. This raises profound questions about human identity, equity, and the potential creation of genetic haves and have-nots. Most countries and the scientific community currently have a moratorium or strict regulations prohibiting heritable human germline editing due to these unresolved ethical and safety concerns. The case of He Jiankui in China, who created the world's first gene-edited babies targeting HIV resistance in 2018, was widely condemned as reckless and unethical. Somatic versus germline distinction. It's crucial to distinguish germline editing from somatic editing. Somatic editing targets non-reproductive cells, like blood cells, lung cells, muscle cells. Changes made are confined to that individual patient and are not passed on to offspring. Most current therapeutic applications focus on somatic editing, which avoids the ethical quagmire of altering the human germline. Eugenics concerns. The specter of eugenics, the idea of improving the human population through controlled breeding or genetic selection, looms large. Could genome editing, especially germline, be misused to enforce narrow definitions of desirable traits or discriminate against individuals with disabilities or undesirable genetic variations? Robust ethical frameworks and societal discussions are essential to prevent misuse. Equity and access. The genetic divide. Cutting-edge therapies are often extremely expensive. Will genome editing cures only be available to the wealthy, exacerbating existing health disparities? Ensuring equitable access is a major societal challenge. Environmental release. Unintended ecological effects. Releasing gene-edited organisms, plants, insects, microbes, into the environment carries ecological risks. Could edited crops outcompete wild relatives, reducing biodiversity? Could gene drives, a powerful application of CRISPR designed to spread a specific edit rapidly through a wild population, e.g., to eliminate malaria-carrying mosquitoes, have unforeseen consequences on ecosystems? Rigorous environmental risk assessments are vital. Biosecurity, the dual use. Dilemma. Like any powerful technology, genome editing could potentially be misused to create biological weapons, engineered pathogens with enhanced virulence or drug resistance. This necessitates careful oversight, responsible research practices, and international dialogue on biosecurity. The future unfolding, editing with wisdom. Genome editing is not a future technology. It's a present reality rapidly evolving. The first CRISPR-based therapies are approved for sickle cell disease and beta thalassemia in the UK and US. Gene-edited crops are being grown, like non-browning mushrooms, high oleic soybean oil, and disease-resistant wheat in trials. The pace of discovery is breathtaking. What's next? Next-gen editors. Scientists are developing ever more precise, efficient, and versatile editors. Base editors that directly change one DNA letter to another without making a double-strand break, reducing risks associated with NHEJ. Prime editors that can make small insertions, deletions, and all base changes more cleanly, and RNA editors that offer temporary, reversible changes. Smarter delivery. Innovations in viral vectors, LNPs, and other methods aim to deliver editors more safely, efficiently, and specifically to target organs or cell types within the body. Epigenome editing therapies, moving beyond the DNA sequence to target the epigenome for diseases where gene activity, not the gene itself, is the problem. Multiplex editing, editing multiple genes simultaneously to address complex diseases or engineer sophisticated traits in crops or microbes. Large-scale DNA writing. Beyond editing, the ability to synthesize and insert entire large DNA constructs opens possibilities for more radical redesigns. The potential of genome editing to alleviate suffering, improve health, enhance food security, and deepen our understanding of life is immense. But wielding this power demands profound responsibility. The journey forward must be guided by rigorous science, unrelenting focus on safety, efficacy, and understanding long-term consequences. Transparent ethics, open, inclusive, and ongoing public and international dialogue about the should we as much as the can we. Defining clear boundaries, especially regarding germline editing and human enhancement. Robust regulation. Developing adaptive, science-based regulatory frameworks that ensure safety without stifling beneficial innovation. Equitable access. Proactive strategies to ensure that the benefits of these technologies are shared globally and do not widen existing inequalities. Education. Fostering public understanding of the science, the potential, and the risks, enabling informed societal decisions. So, here we are. We hold the pencil, not a metaphorical one, but a real molecular tool capable of erasing typos and rewriting sentences in the grand instruction manual of life. We peered into the cell's nucleus, deciphered the four-letter code, and discovered in the unlikeliest of places, a bacterial battleground, the molecular scalpel that makes editing possible. We've seen the intricate dance of cutting and repair, the dazzling promise of curing genetic diseases, creating climate-resilient crops, and unlocking biological secrets. And we've also stared into the ethical abyss, recognizing the risks of unintended consequences, the peril of altering future generations without their consent, and the potential for misuse and inequality. Genome editing is not magic. It's hard-won science, still young and learning to walk. It's a tool, arguably one of the most powerful humanities has ever grasped. And like any powerful tool, fire, electricity, nuclear fission, its impact depends entirely on the wisdom, foresight, and ethics of those who wield it. Will we use it primarily to heal, to nourish, to understand? Or will hubris and haste lead us down paths that cause harm or create new forms of injustice? The story of genome editing isn't finished. It's being written right now in laboratories and ethics committees, in farm fields and hospital wards, in public forums and international treaties. It demands not just scientific brilliance, but also deep compassion, careful deliberation, and a shared commitment to using this knowledge for the greater good. It challenges us to define what kind of future we want to build, not just for ourselves, but for generations to come and for the living world we share. The potential is breathtaking. Imagine a world free from the shadow of Huntington's disease or sickle cell anemia. Imagine staple crops thriving in drought-stricken regions, ending famine. Imagine personalized cancer cures tailored to an individual's unique tumor. This is the horizon genome editing offers. But let us walk towards that horizon, not with reckless abandon, but with measured steps, clear eyes, open dialogue, and an unwavering commitment to do no harm. Let us edit with humility, guided by the profound understanding that we are not just rewriting code, we are reshaping the very fabric of biology, and with it, the future of life on Earth. The pencil is in our hands. Let's write wisely. Bye-bye, and good night.